Yeah, so my name is Jose Lugo-Martinez, and I'm a professor in the Bio Department. And so the big goal of my lab is to sort of develop the next generation of computational approaches that would actually sort of enable us to accelerate scientific discovery through the use of automated science. And here, I just have a very brief overview of what automated science means. And basically, what the idea is that you have a closed loop where you have robots that are actually generating data. And this data is actually being interpreted mostly through machine learning methods in order to make predictions. And then we have some sort of AI that drives what will be the best experiments to do next. And then they just pass this information to the robots that are actually going to perform this new set of experiments. And so my lab tries to get involved in all aspects of this. However, in practice, it is very possible that you don't have a closed loop system that the one I was showing at first, where you may have some manual data generation. So you have some collaborators that are actually you're going to pass the information, or they're going to generate data for you that you are going to model, and then that you're going to give them recommendation of experiments to do next. And so this is sort of the more practical view or more general view of the kinds of sort of setup or pathways that you can have under an automated science framework. And to put this in context, so I'm a computational biologist. So the kinds of problems that I work with are, inspired by biology. And one of the main biological questions that I'm interested in is in trying to understand the spatial-temporal dynamics of protein complexes. And here, just as a very brief example, I'm showing a protein complex. What I refer to as a protein complex is just a group of proteins that cooperate to perform some biological function. And the critical thing here is, as I'm showing here with this picture, is that we have a protein complex. And the important thing to note here is that some proteins actually directly interact, but there are others that don't directly interact. However, they together cooperate to perform a biological function. And if you want, you can just sort of extract these in this representation where you have all the proteins sort of connected within this circle that represent the complex. And sort of one of the main or underlying hypotheses that we have is that if we can actually somehow try to understand the mechanisms behind these protein complexes, then maybe this will enable us to actually understand the behavior of a cell system. So this is the underlying sort of idea. However, one of the main challenges is that we actually, in order to do that, we need data, and we need some more accurate maps of protein complexes. And what happens is, even for humans, this is largely unexplored. And so one of the main projects that we do in the lab is we develop methods based on existing data to try to not only use different mathematical models, in this case hypergraphs, but also combine these with different types of data sources, heterogeneous data sources, in order to predict potentially novel protein complexes. And in addition to this, then we use AI to select what experiments to do next. And we actually program, configure, and use robotic experiments to actually perform these experiments. And you can imagine that in a set where you have multiple choices of experiments, you want to optimize what would be the best experiment to do in order to try to find supporting evidence to whether the prediction is actually something that we are observing in nature. And so these are sort of the main problems that we work in the lab. If any of these have interest to you, feel free to reach out and contact me. I'm either in my office in GSC 7411, or you can reach out to my email, which is listed here. Thank you.